of the drug development and the clinical trial design. This is the fourth part of the lecture one, which is a drug development and the clinical trial design. We will, discuss the phases of drug development and the clinical design. Is structured across the multiple phases, each requiring specific study designs to systematically evaluate the safety, efficacy, and overall benefit risk profile of therapeutic intervention. So each study or each phase require a specific study design, and we can notice that through the discussion, and that is just to evaluate the safety, efficacy, and overall benefit risk profile of therapeutic intervention. The choice of a study design is informed by the research objectives at each stage, with a strong emphasis on generating reliable and generalizable data that we can say like internal validity and external validity. The drug development process is structured into four phases. When we just make a new drug and we wanted to, market this drug, or we just send this drug to the market, it structures into four phases, each with its own objectives, key consideration, study designs, and the goals. These phases help ensure the safety, efficacy, and long term effectiveness of a new therapeutic interventions. And here we will break down each phase, we will understand each phase. So let's start with the phase 1, which is known as a human pharmacology. And from its name, you can notice that it will be about the pharmacokinetics and the thermodynamics. So what is the objective? Each phase we will discuss what is the objective, what is the key consideration, and what is the or what are the study designs. Okay, so the objective of the first one, which is known as a human pharmacology, it assesses the safety, tolerability, pharmacokinetics, and thermodynamics of a drug in humans. And the pharmacokinetics, IDMA, thermodynamics, the mechanism of the drug, identify the maximum tolerated dose and establish an initial safety profile. So the objectives, the first one is assess the safety, tolerability, pharmacokinetic, thermodynamic of a drug in a human, and identify the maximum tolerated dose. That's mean the maximum dose that can the human tolerated it, and establish an initial safety profile. What are the key considerations? Studies generally involve healthy volunteers, not patient, or maybe patient. Studies generally involve healthy volunteers or patient for high toxicity drug like oncology agents. About what is the design, it's non-randomized open label design. That's mean what we mean exactly by the open label design I discussed before. It means the participant and the researchers know the treatment to monitor the safety closely. Study designs like single ascending dose. Smaller groups receive an increasing doses until dose limiting toxicities are absorbed. So they will receive an increasing dose till the dose limiting toxicity, but it's just one dose, or the dose is increasing. Okay, so the single ascending dose is smaller groups receive increasing doses until dose limiting toxicities are absorbed. Multiple ascending dose, participants receive multiple doses over time to assess cumulative effects. Okay, because if there is a multiple doses with a shortness of time interval, that is lead to the cumulative effects. And we have the food effect studies, examine how food influences drug absorption and bioavailability. Okay, and the other things is micro dosing studies, like administer subtherapeutic doses to assess the pharmacokinetics without pharmacological effects. Phase 2 is therapeutic exploratory studies. The objectives is optimize dosing regimens, evaluate preliminary efficacy, and assess further safety in a defined patient population. That mean find the best amount of the medicine to give. That is like the other meaning to know what he exactly means by optimize dosing regimen, evaluate preliminary efficacy, and assess the further safety in a defined patient population. It means find the best amount of the medicine to give, sees if it works well, and check if it's safe for a specific group of patients. The findings help design phase 3 trials and refine the treatment strategies. About the key consideration, large sample sizes than the phase 1 but is still relatively small. Double blind designs, that mean the participant and researcher they don't know what is a treatment to reduce the bias and improve the reliability of the results. The study designs is like randomized control trials, compare different doses or treatment option to minimize the bias and establish the causal relationships. Adaptive designs modify the trial based on the interim data. That means it's temporary data such as a dose adjustment or sample size re-estimation. That's mean exactly if the really results or early results data shows something important, the trial can be adjusted to improve it. Like not just waiting to in the clinical trial and then improve the results. No, just we observe that, and if there is early results or data show something important, then okay, we adjust it during this trial. We just not waiting to in this trial and then improve it. No, we can improve it while it happen. Biomarker enriched designs, that mean group participants based on a specific biological markers that help predict how well they will respond to the treatment. And we have also cohort expansion studies used in oncology to expand initial safety data into large cohorts to gather more efficacy data. That's mean for the cancer research, after testing a treatment in a smaller group of patients to check the safety, we can expand it to more patients to see if the treatment works well or not. Just like expansions, we like increase the number of the participants in the group so we can see how the treatment is is safe and how it's work well. um, Phase 3, it's therapeutic confirmatory studies, and the objective confirm the efficacy. I mean, efficacy, does the treatment work, and safety, is the treatment safe. That is the difference between the efficacy and the safety, and we just, from its name, confirmatory. That's mean we just confirm the efficacy, does the treatment work, and the safety, is the treatment safe of the drug in a larger scale studies, and provide definitive evidence for regularity approval. Provide data to support FDA, which is Food and Drug Administration Organization, and EMA, European Medicines Agency submissions and approval. About the key considerations, multi-center often international trials require substantial resources, uses of blinding and placebo control to ensure rigorous evidence, strong evidence. The study designs, parallel group randomized control trials, participant randomly assigned to either a treatment or control group, the most common design in a phase 3. Non-inferiority trials designed to show that the new treatment is not worse than an existing therapy. And factorial designs, this multiple intervention simultaneously evaluating combined therapy effects. And event driven or driven designs, instead of going with a fixed time based design like just waiting 2 years, the trial is driven by the number of important events that could be happen. I mean, we can in the design if there is happen like heart attack or cancer progression. And the phase 4, which is known as a post marketing surveillance, Objective monitor ongoing safety and effectiveness of a drug after market approval, assess long-term outcomes, identify rare adverse effects or events, and explore new indication or combination because it's for the market, so we can observe more data. About the key consideration, real world data becomes increasingly important, often leveraging electronic health records or healthcare system for large scale studies and continual pharmacovigilance and risk management to maintain the drug's benefit risk profile. About the study design, observational studies cohort and case control studies to track real world safety and effectiveness. Registry studies collect the data from a large population over time to understand how the drug performs across the subgroups, and we have randomized pragmatic trials, and I mean exactly by the pragmatic it's more practical. Compare the new drug with the standards treatments in routine clinical practice to assess the real world effectiveness. I mean pragmatic means that these trials are designed to be more practical and closer to how the treatment are actually used in everyday clinical practice. And now we can go to the fifth topic, on the final topic in our lecture, is formalizing the study design in clinical study protocols. So the clinical study protocol is a critical document that meticulously, that's mean a very accurate, outlines every aspect of a clinical study, serving as a comprehensive roadmap of its conduct, adhering to the International Council of Harmonization guidelines. The clinical study protocol ensures that the study is scientifically robust, ethically sound, and compliant with the regulatory requirements. It provides a transparent and detailed framework that facilitates a reproducibility and maximize the credibility of the research findings. Now we go to the key component of clinical study protocol. The first one is the study objectives and hypothesis, design and methodology and statistical analysis plan, ethical consideration, and that's it. So the first one is study objectives and hypothesis. Primary objectives clearly define the main research question, and the primary hypothesis typically linked to the study most crucial outcome. Secondary objectives provide additional research questions or explore secondary effects of the intervention. Exploratory objectives identify a new hypothesis or research areas for the future investigation. The hypothesis should describe the expected direction and the magnitude of the intervention's effect. The second thing is the design and methodology. Study type specify whether the study is interventional or observational, and state its phase 1, 2, 3, or 4. Intervention models outline the structure, like if it's parallel design, crossover design, factorial design, and describe the treatment arms and control groups. Randomization explain randomization procedures, allocation, concealment, and randomization ratios. Mention any stratifications or blocking to minimize the selection bias. Blinding detail whether the study is open label, single blind, or double blind, and how blinding will be maintained. Outcome measures define primary and secondary outcome measures, explaining how they will be assessed, timing of assessment, and the measurement methods. Ensure that the outcome are clinically relevant and sensitive to the intervention effects. 3. Statistical analysis plan. Primary and secondary hypothesis testing, like specify statistical test for hypothesis testing and significant level. There is a 5% chance that the result you found happened by the random chance and not because the treatment or effect you are testing. So discuss the adjustment for multiple comparisons if needed. Sample size calculation justify the sample size required to detect a meaningful effect and include consideration like expected effect size, power, significance level, and expected attribution rates. Handling missing data outline strategies for dealing with missing or incomplete data, such as imputation methods, for example. In term analysis and stopping rules, that means it's the temporary analysis, if applicable, define the rules of the interim analysis and the criteria for halting the study every early due to the efficacy or safety concerns. So we can make that is temporarily. Subgroup analysis describe any planned subgroup analysis, providing rational and statistical consideration for this. Finally, the ethical consideration, regularity, compliance, ensure adherence to good clinical practice, the correlation of Helsinki and other regularity standards, and that it will be the second lecture. It will be talk about these regulations. Informed consent explain how the participants will be informed about the study, including its risk, benefits, and how consent will be obtained. Participant safety describe the safety monitoring plan, including the role of the data safety monitoring reports, adverse event reports, and how risk will be mitigated. Vulnerable populations, if applicable, outline protections for vulnerable groups such as children, pregnant women, and individuals with impaired decision making capacity. And the role of CSP in ensuring scientific and ethical integrity. Um, it ensures that the clinical trial is designed to achieve scientifically robust results while protecting the rights and the safety of participants. It serves as a detailed blueprint for study execution, helping to ensure that scientific rigor and ethical responsibility. So the first one is the study design addresses the research questions accurately and effectively. Potential biases like selection bias, performance bias are minimized. The data collection and analysis are planned to be reproducible and transparent. About the ethical responsibility, participants' welfare is prioritized, ensuring risks are minimized and informed consent is obtained. Ethical standards are upheld by complying with the relevant guidelines, protecting vulnerable population and ensuring participant autonomy. And finally, translating observation into generalizable knowledge. The CSP enable researchers to translate limited controlled observation into broader generalizable clinical knowledge by adhering to the guidelines like ICH E6R2, which means good clinical practice. The protocol ensures that the study provide high quality evidence that can inform clinical practice and future research. The structured approach facilitates regularity review and enhances scientific credibility. So by this we end the part 2 of the lecture 1. I know it's so tough, I can't explain more, it's just we make it easy as I can. So we finish now with the clinical trials, the lecture 1. Cheers.